Now, let us proceed to our next lesson, 18.2, Transmission Electron Microscopy. So what is a transmission electron microscope? Transmission electron microscopes, or TEMs, form images using electron that are transmitted through a specimen. The transmission electron microscope can provide vastly greater resolution than the light microscope, and it can magnify objects up to 2 million times. So it is no wonder that transmission electron microscopes have become so valuable within the biological and medical fields. Now the question is, how do transmission electron microscopes work? So TEMs employ a high voltage electron beam in order to create an image. An electron gun at the top of a TEM, this one. Alright. It emits electrons that travel through the microscope's vacuum tube. Rather than having a glass lens focusing the light, the transmission electron microscope employs an electromagnetic lens which focuses the electrons into a very fine beam. This beam then passes through the specimen, which is very thin, and the electrons either scatter or hit a fluorescent screen at the bottom of the microscope. An image of the specimen with its assorted parts, shown in different shades according to its density, appears on the screen. And this image can then be studied directly within the transmission electron microscope or photographed. Alright, so although transmission electron microscopes and light microscopes operate on the same basic principles, there are several differences between the two. And the main difference is that transmission electron microscopes use electrons rather than light in order to magnify images. The power of the light microscope is limited by the wavelength of light and can magnify something up to 2,000 times. Electron microscopes, on the other hand, can produce much more highly magnified images because the beam of electrons has a smaller wavelength, which creates images of higher resolution. Now, this illustration that you can see on your screen, on the left side, this one, this illustration is taken by a light microscope. So this is a cotton stem, and this area, kaning circle, this is the phloem tissue. On the right side, this illustration, this is an enlarged image of cotton phloem tissue. And this was taken using transmission electron microscopes. So as you can see, mas klaro ang kuha sa transmission electron microscope as compared with light microscope. Now, this illustration shows the preparation of a specimen for observation in the electron microscope. As with the light microscope, tissues to be examined in the electron microscope must be fixed, embedded, and sectioned. Fixation of tissue for electron microscopy is much more critical, meaning lisod, than for light microscopy because the sections are subjected to much greater scrutiny. So it means that it needs critical observation or critical investigation. A fixative must stop the living cell without significantly altering the structure of the cell. And the most common fixatives for electron microscopy are glutaraldehyde and osmium tetroxide. Now, once the tissue has been fixed, the water is removed by dehydration in alcohol, and the tissue spaces are filled with a material that supports tissue sectioning. Tissues to be sectioned for electron microscopy are usually embedded in epoxy resins such as epon or araldite. Now, there are various methods used to examine or to study the specimens using transmission electron microscopes. So we have here cryofixation and the use of frozen specimens. Cells and tissues do not have to be fixed with chemicals and embedded in plastic resins in order to be observed with the electron microscope. Alternatively, cells and tissues can be rapidly frozen. Because cryofixation accomplishes these goals without altering the cell's macromolecules, it is less likely to lead to the formation of artifacts. So when we say artifacts, this is the changes brought in the tissue as a result of human activity. The major difficulty using cryofixation is the formation of ice crystals which grow outward from sites where nucleation occurs. As an ice crystal grows, it destroys the fragile contents of the cell in which it develops. So the best way or the best method for that is to avoid ice crystal formation by freezing the specimen so rapidly that crystals don't have time to grow. Another method for transmission electron microscopy is what we call as negative staining. The electron microscope is also well suited for examining very small particulate materials such as viruses, ribosomes, multi-subunit enzymes, cytoskeletal elements, and protein complexes. The shapes of individuals' protein and nucleic acids can also be resolved as long as they are made to have sufficient contrast from their surroundings. And one of the best ways to make such substances visible is to employ negative staining procedures in which heavy metal deposits are collected everywhere on a specimen grid except where the particles are present. As a result, the specimen stands out by its relative brightness on the viewing screen. Now let us tackle about shadow casting. Another technique to visualize isolated particles is to have the objects cast shadows. The grids containing the specimens are placed in a sealed chamber, which is then evacuated by vacuum pump. The chamber contains a filament composed of a heavy metal together with carbon. The filament is heated to high temperature, causing it to evaporate and deposit a metallic coat over accessible surfaces within the chamber. As a result, the metal is deposited on those surfaces facing the filament, while the opposite surfaces of the particles in the grid space in their shadow remain uncoated. When the grid is viewed in the electron microscope, the areas in shadow appear bright on the viewing screen, whereas the metal-coated regions appear dark. The convention for illustrating shadowed specimens is to print a negative image in which the particle appears illuminated by a bright, white light with a dark shadow cast by the particle. The technique provides excellent contrast and produces a three-dimensional effect. Lastly, we have freeze fracture, replication, and freeze etching. So, freeze fracture describes the technique of breaking a frozen specimen to reveal internal structures. Freeze etching, on the other hand, is defined as the sublimation of surface ice under vacuum to reveal details of the fractured face that were originally hidden. So that would be all for the lesson Transmission Electron Microscopes.